Kochani uczestnicy, szanowna komisja, ze względu na to, że nie zjawiły się żadne osoby z zagranicy, nasza sesja zostanie przeprowadzona w języku polskim. Szanowna komisjo, drodzy uczestnicy, słuchacze, panie i panowie, w imieniu Studenckiego Towarzystwa Naukowego Śląskiego Uniwersytetu Medycznego w Katowicach chciałabym serdecznie podziękować za przybycie i udział w SIMC 2025. Nazywam się Julia Herbuś i mam zaszczyt pełnić rolę koordynatora sesji Basic Science and Biotechnology and Biological Engineering. Serdecznie witam szanowną komisję. Bardzo dziękuję za przybycie i chęć tutaj ocenienia naszych prac. Bardzo też dziękuję komisji, która jest teraz na Zoomie zdalnie. To naprawdę niezwykłe, że udało nam się w komisji zebrać prowadzących z aż 3 uczelni. Na początek chciałabym wyjaśnić zasady obowiązujące podczas sesji. Kolejność prezentacji odpowiada kolejności zamieszczonej w książce abstraktów. Maksymalny czas na prezentację to 7 minut oraz 3 minuty na dyskusję. Za przekroczenie czasu będą naliczone ujemne punkty. Jeśli kogoś nie ma na sali, kiedy zostaje wywołana jego sesja, praca zostaje przesunięta na koniec sesji. Szanowna komisjo, pragnę uprzejmie przypomnieć, że w przypadku, gdy są Państwo opiekunem naukowym prezentowanej pracy, nie powinni Państwo jej oceniać. W takim przypadku proszę skreślić numer pracy na karcie oceny. Pozostali członkowie tej samej kliniki mogą oceniać daną prezentację. Oceny będą weryfikowane. Wszystkie prace oraz dyskusje muszą być prowadzone w języku angielskim, jeżeli w takim języku zostanie przeprowadzona prezentacja. Jeżeli zaś decydujecie się na wygłoszenie prezentacji w języku polskim, dyskusja też może się odbyć w języku polskim. Uprzejmie proszę komisję o ocenę według następujących kryteriów. Wartość naukowa, punkt 1.5, sposób prezentacji, punkt 1.5, dyskusja, punkt 1.5 oraz wkład własny, punkt 1.5. Po zakończeniu sesji prosimy o zwrot kart oceny tutaj do koordynatora albo do mojej asystentki. Karty zostaną wrzucone do naszego elektronicznego systemu, który automatycznie wyliczy punkty i wyłoni zwycięzców sesji. Czy wszystkie zasady sesji są jasne? Czy uczestnicy albo komisja mają jakieś pytania? Tak, słucham? Tak, ze względu na to, że według regulaminu ta sesja nie jest obligatoryjnie w języku ani angielskim, ani polskim, można przedstawiać zarówno w jednym, jak i drugim języku. Tylko prezentacja musi być obowiązkowa w języku angielskim, co sprawdziłam, wszystkie prezentacje spełniają ten wymóg. Tylko tak jak mówię, jeżeli prezentuje się w języku angielskim, dyskusja również odbywa się w języku angielskim. I uczulam Was, uczestnicy, jeżeli komisja zada Wam pytanie w języku polskim, a prezentowaliście po angielsku, powinniście odpowiedzieć po angielsku. Czy teraz wszystko jasne i możemy zaczynać? Dobrze, nikt nie ma pytań. W takim razie zapraszam tutaj na scenę reprezentanta pierwszej pracy. Jest na sali Kacper Odziomek od pracy Evaluation of Hydrocortisone Delivery. Okej, jesteś zdalnie, dziękuję Ci bardzo. W takim razie, skoro jesteś zdalnie, proszę Cię o udostępnienie ekranu, ponieważ w tym wypadku to Ty udostępniasz swoją prezentację.
Mhm. Wszystko widać?
Tak, widać, tylko prezentacja nie jest na pełnym ekranie. Jakbym mogła prosić, żeby tutaj postarać się ją włączyć mimo wszystko na pełen ekran. Tutaj w prawym rogu masz taki znaczek z stroną i jak to rozwiniesz, to powinieneś mieć, o właśnie, tryb właśnie prezentacji.
Spróbuję udostępnić tylko okno, bo tutaj
Już
chyba...
wszystko widać teraz.
Tak?
Dobrze. Tak, można zaczynać.
Dobrze. To w takim razie ja nazywam się Kacper Odziomek. Jestem studentem studiów magisterskich na Politechnice Krakowskiej imienia Tadeusza Kościuszki. Jest mi bardzo miło, że mogę zaprezentować swoją pracę na temat oceny właściwości hydrożelowych systemów dostarczania hydrokortyzonu w kierunku leczenia terapii, w kierunku leczenia łuszczycy. E, Jeśli chodzi o łuszczycę, tylko, no to tak jak wiadomo, następuje dosyć duży rozwój w kierunku nowoczesnych terapii, natomiast całkowite jej wyleczenie pozostaje niemożliwe. Też skuteczność takich tradycyjnych preparatów jest ograniczona przez skutki uboczne lub tłustość po prostu tych formulacji, co może również zmniejszać skuteczność, ponieważ tutaj ważne jest przestrzeganie tych zaleceń przez pacjentów. I tutaj moje badania dotyczą właśnie hydrożelowych biomateriałów, które charakteryzują się przedłużonym i kontrolowanym uwalnianiem substancji czynnych, w tym przypadku hydrokortyzonu, co powinno zwiększyć tą skuteczność terapii. Jeśli chodzi o metodykę, postaram się to tak szybko streścić. Na początku polimerowy nanonośnik został syntezowany poprzez polimeryzację emulsyjną z N-izopropyloakrylamidu. Następnie enkapsulacja hydrokortyzonu w tym nanoczułym nanonośniku została przeprowadzona przez intensywne mieszanie i taki nanonośnik został następnie zliofilizowany. Natomiast aby otrzymać biomateriały wykonane przede wszystkim z polialkoholu winylowego, alginianu sodu i ekstraktu z aloesu, przeprowadzono sieciowanie chemiczne, używając do tego dodatkowo gliceryny i czynnika sieciującego. Mm, I tutaj średni rozmiar cząstek tego nanonośnika został określony. Wynosił on pomiędzy 125 a 137 nanometrów, natomiast współczynnik polidyspersyjności pomiędzy 0,17 a 0,20, więc ta enkapsulacja hydrokortyzonu nie spowodowała żadnych znacznych zmian w układzie. Mm, Stopień usieciowania biomateriału wahał się między 61 a 70%, natomiast stopień pęcznienia pomiędzy 184 a 261%. Pomiary pH i przewodnictwa potwierdziły, że biomateriały te są stabilne w szerokim zakresie pH. Struktura chemiczna biomateriału została potwierdzona poprzez analizę FTR. I też integralność strukturalna była dobrze zachowana. Tutaj sugeruje to, że wprowadzanie nanonośnika związanego z hydrokortyzonem do biomateriałów nie zaburza generalnie całej ogólnej morfologii próbek. Na rysunku 9D po prawej stronie można zobaczyć też cząsteczki leku. Natomiast to, co jest ważne, to udało się uzyskać to przedłużone i kontrolowane uwalnianie do 7 dni. Mm, Uwalnianie zachodziło zgodnie z kinetyką dyfuzji Ficana, czyli taką typową kinetyką bez żadnego wpływu pęcznienia jako głównego czynnika kontrolującego ten proces, a kinetyka uwalniania najlepiej charakteryzował model Korsmeria-Pepasa. Jeśli chodzi o cytotoksyczność, oczywiście zbadano tutaj próbkę, która wykazywała najlepsze właściwości fizykochemiczne. Zbadano ją na modelu 3D łuszczycy przeżywalność komórek po 24 godzinach z taką próbką wykazywała wartość około 87%. Natomiast przebadano również ten model pod kątem właśnie ekspresji, hamowania ekspresji genów typowych dla hiperproliferacji i stanów zapalnych w łuszczycy. I tutaj na rysunku 13 można zauważyć, że w zależności od rodzaju danego biomarkera, to hamowanie ekspresji było różne. Natomiast to, co jest ważne, to to, że ta próbka faktycznie hamowała tą ekspresję, czyli tak naprawdę ta skuteczność biologiczna została potwierdzona. Także podsumowując, otrzymano biomateriały hydrożelowe o wysokim potencjale do komercjalizacji jako opatrunków łagodzących objawy łuszczycy. Część biologiczna została wykonana w ramach projektu Future Lab. I to wszystko. Dziękuję za uwagę. Jeśli mają Państwo jakieś pytania do mnie, to proszę.
Bardzo dziękujemy za tą prezentację i zachęcam komisję do zadawania pytań. Tutaj komisja, czy stacjonarna, czy zdalna, ma jakieś pytania do pracy?
Dzień dobry, witam serdecznie. Jeśli mogę pozwolić sobie na zadanie pytania, chciałbym zapytać
Dobry.
o Pana wkład własny w wykonanie tej pracy. Jaki był Pana udział, jaki był udział poszczególnych członków Pana zespołu i w jaki sposób właśnie to badanie Państwo przeprowadzili?
Mhm. Wszystkie badania, które były wykonywane przeze mnie, natomiast część biologiczna była wykonywana w ramach stażu badawczego na Uniwersytecie Śląskim pod opieką naukową dr Katarzyny Malarz i profesor Anny Mrozek-Wilczkiewicz. Także no, moja praca to była synteza biomateriałów, następnie ich charakterystyka fizykochemiczna, strukturalna, morfologiczna, badania nad profilem i kinetyką uwalniania substancji czynnych. I tak jak mówię, ta część biologiczna też również była wykonywana, ale w ramach projektu razem z koleżanką wykonywaliśmy te testy.
Bardzo dziękujemy. Czy są jeszcze jakieś pytania? Jeśli nie ma więcej pytań, bardzo dziękujemy za prezentację i można zakończyć udostępnianie ekranu.
Dziękuję bardzo. Do widzenia.
Is the representative of the second presentation with us on Zoom? Okej. W takim razie druga praca na ten moment spada na ostatnie miejsce na naszej liście. Przechodzimy do pracy numer 3. I tutaj będzie, czy jest z nami Ignacy Berendt? To zapraszam w takim razie.
So, hello everyone, my name is Ignacy and I study medical informatics at Wrocław University of Science and Technology. And today I would like to present results of my research on influence of sex and breathing on autonomic nervous system during controlled breathing using transfer entropy algorithm. So myself, I'm also leader of scientific lab Signum at my university, where we are dealing with IT in medicine. So we are very open for different collaborations. Also, this is part of the project Automatic at BrainLab Research Group, where we are dealing with signal analysis and also, among others, with autonomic nervous system. So this is partially also our motivation for this work. And what we know from the literature is that controlled breathing modulates autonomic nervous system response. And also there is a thing called baroreflex. This is a negative loop that in response of increasing arterial blood pressure causes slower heartbeat. And also in case of lower arterial pressure, it causes faster heartbeat. So there is a causality between heart period and systolic arterial pressure. And also we believe that this kind of research on healthy individuals will help us to understand what happens in pathological scenarios on clinical patients, especially under mechanical ventilation in this case. So what is our aim? Our aim is to test transfer entropy algorithm. And this is a model-free measure of directed information flow. And also what is good about this algorithm is that it also helps us to identify like nonlinear dynamics in our system. And also want to investigate how sex and resting breathing level influence the response of the autonomic nervous system during controlled breathing. So what materials and methods we used? So we had 24 healthy students who participated in this study, and we were measuring two signals, arterial blood pressure and also respiratory rate. And the protocol was such that volunteers were breathing at first at resting, and then during controlled breathing at 6, 10, and 15 breaths per minute, guided by metronome. And every of this measure was 5 minutes long. And this is also setup of our measurement setup. Yep. And this is how our signals were looking like. This sample of arterial blood pressure at different breaths per minute after cleaning that we used later in the algorithm. And we wanted to derive two signals from our arterial blood pressure. It was systolic arterial pressure, so this is simply just following peaks in our arterial blood pressure. And also we wanted to get heart period, so this is simply reverse of distance between two following peaks. So we got two signals from our arterial blood pressure. And to understand the transfer entropy, we have to take 2 steps back. So first question is, what is entropy? We are using information theory definition, and simply we can state it as amount of information in the system. And then to build transfer entropy, we need to use conditional entropy. And this is simply amount of information left in the system, while we know also, in our case, other signal y. And using that, we can build transfer entropy. It's actually a special case of conditional entropy. And this is reduction of uncertainty of xt plus 1 when we include past values of y. So amount of information transferred from system y to system x. And this should be directional, so direction matters. And this is what we see here. So transfer entropy from heart period to systolic arterial pressure, there is no significant difference between our breathing groups. But if we take direction of systolic arterial pressure to heart period, what happens in baroreflex, then we see that the transfer entropy is increasing while the breathing frequency is increasing, and this is something that we expect. So then we wanted to ask 2 questions. Does sex matter? This is the first question, and we see that the only difference that was significant was at resting breathing for transfer entropy from sub to HP. And the second question was that does it matter if our volunteers, if they are breathing normally at a higher pace or at slower pace? In our case, the threshold we set at 15 breaths per minute, and we see that it doesn't matter, it doesn't have significant difference. So for the conclusions, as I mentioned, sex significantly impacted only transfer entropy for resting breathing. Resting respiration level influence might be also neglected. And obviously, we want to perform further research to on the patients in clinical scenarios, so with pathologies. And that would be it.
Tym razem ja się zapytam o wkład własny pana w przedstawione badania.
Okay, so if like from the beginning, the data wasn't collected by me. Actually, I was one of the people on whom the study was done. So it was all acquired by my tutor and her group. So I had already data and it was already cleaned. So my work was to implement this transfer entropy algorithm. I was using Python for that, and to put all the data do the calculations, do plots, and also with help of my tutor do the statistical analysis. I was using repeated measurements, ANOVA for that.
Okay, thank you.
Czy są jeszcze jakieś pytania tutaj z Zooma albo od uczestników? Czy chcielibyście zadać jakieś pytanie naszemu reprezentantowi? Dobrze, jeśli nie ma więcej pytań, ja bardzo dziękuję za prezentację. So, is the representative of the next presentation is with us?
Yes, I think it's me, right?
Right. Thank you.
Okay, I'll turn on the camera and I'll share my screen, so let me know, please, if you can see it.
Okay.
Now?
Yes, we see everything.
Okay, perfect. So, first of all, good morning, everyone, and thank you for the opportunity to present our research here today. So my name is Daria, and let's begin then. Yeah, 1 second. So the topic of our research is bioinformatics screening of deleterious, non-synonymous SNVs in human gene GATA1. So let's start with the background. So the GAT1 gene encodes a transcription factor that binds to DNA sequence of guanine, adenine, thymine, and adenine. And it's crucial for hematopoiesis. And therefore, mutations in GAT1 are linked to disorders like Diamond-Black vein anemia, thrombocytopenia, and acute megakaryoblast leukemia in children with Down syndrome. So GAT1 alterations are also seen in leukemia relapses after chemotherapy. And while single nucleotide variants, SNVs account for about 90% of genetic variation, and research on SNVs in GAT1 is limited. So the aims of the study was to evaluate the non-synonymous SNVs in the GAT1 gene, focusing on their structural and functional and clinical importance. So the study included 494 missense SNVs in the GAT1 genes, retrieved from the UniProt database, and we assessed each variant structural and functional effects by 6 predictors, while 3 tools assessed their impact on protein stability. So variants were classified as pathogenic if all tools employed in the study predicted them to be deleterious and decreasing protein stability. So the evolutionary conservation of amino acids in the GAT1 transcription factor was analyzed by the conserv server. So to evaluate SNV's potential pathogenicity, we used 6 different bioinformatics tools that predict the impact of amino acid substitutions on protein function. And the results of the predictions are clearly illustrated in this bar chart, and you can see the services that we used. And a minimum number of deleterious SNVs were detected by SNPs and GO, and you can see it here, while the largest one was found by Phantom, and you can see it in this. So after collecting predictions from all 6 tools, we performed a consensus analysis to assess how consistently each SNV was identified as potentially pathogenic, and the results of this consensus analysis are shown in this pie chart. So variants that were predicted as deleterious by all 6 tools were labeled pathogenic, as shown here, and we identify 71 pathogenic SNVs. Interestingly, only 68 of all SNVs analyzed had been previously annotated as pathogenic in clinical or bioinformatical databases, but most of them were out of the list of pathogenic SNVs. And thus, 53 additional SNVs were recognized as pathogenic. So in addition to structural and functional predictions, we also evaluated the potential impact of SNVs on protein stability. So we used 3 in silico tools to assess protein stability. So that's iMutant2, MuPro, and iStable, as shown in this bar chart. And all 3 tools predicted nearly identical results with 346 or 347 SNVs decreasing protein stability. You can see it here. So when combining this and previous prediction, a total of 266 SNVs were consistently classified as destabilizing. So among them, 33 were already annotated in clinical databases as pathogenic or likely pathogenic, and 39 destabilizing. SNVs were newly identified and representing over 60% of all new deleterious variants. And also 55 SNVs across 44 amino acid positions were predicted to be both pathogenic and destabilizing, highlighting them as a high confidence candidates for clinical significance. So to understand the structural importance of SNVs, we analyzed the evolutionary conservation of amino acid residues in GAT1, and highly conserved regions identicate functionally critical sites where mutations are damaging. And our analysis classified 26.4% of GAT1 amino acids as highly conserved, and notably 64.8 of newly predicted pathogenic SNVs were found to be highly conservative, supporting their clinical significance. And analysis showed that many conserved residues cluster between amino acid positions 198 and 302, you can see by this region, where zinc fingers domains are located, essential for DNA binding and interaction with transcription cofactors. So mutations in this region may impair the regulation of GAT1 target gene expression, explaining the links to blood disorders. So on this slide, you can see conclusions that have been announced during the report, and these findings highlight the value of integrating sequence-based structural and evolutionary analysis in SNV screening. Further validation could support targeted sequencing and early diagnosis of hematological diseases linked to GAT1 gene mutations. And thank you for attention.
Thank you for your presentation. And now I would like to invite jury and all participants to ask questions and discuss the present paper.
Hello, thank you very much for your presentation. It was very interesting. Thank you very much for it. And I would like to ask you about your future plans. What do you think? Is it possible to transfer this analysis into practical or even commercial use? Where you can see this project in the next 2 or 3 years?
I'm only first-year student, so of course, I'm not sure how we can use this as commercial in the future. I mean, like for me, like in 2 years, because I'm only a student now. But I find this topic very interesting, and of course, we know about genetic engineering technologies now, and we can we can treat beta thalassemia and other blood disorders. So I think it's like a good start to start thinking how we can use this as a potential to maybe achieve something new in treatment, diamond blackfan anemia and other disorders linked to GAT1. And also this study shows us interactions between genes. So I think it's valuable, yeah, for future research. But for now, I can't really say how I will use this research like in 2 years because I'm only a student.
Absolutely, I totally agree with you. It's a fantastic start, and thank you very much for this presentation and congratulations. Because if somebody can do something like this in his or her first year, it's amazing. Thank you very much.
Thank you so much. Thank you.
Are there any more questions? Okay, so thank you, Daria.
Thank you.
W takim razie zapraszam przedstawiciela kolejnej pracy tutaj do nas na scenę.
Good morning, my name is Agata Gut, and it's a great pleasure to be here today to talk to you about the association of entropy measure with brain-specific serum biomarkers in traumatic brain injury. This study was supported by the National Science Center and it's a part of a Center TBI project. The brain itself can be viewed as a complex organ. Therefore, when it's damaged, the response may be also multi-phased. The traumatic brain injury is caused by an external force, and it is responsible for the majority of trauma-related deaths. On the one hand, the traumatic brain injury may cause the stimulation of the autonomic nervous system, and in this way, in the sympathetic pathway, the catecholamine release occurs, and that results in the hypertension and the increase of the intracranial pressure. On another hand, the TBI may lead to the disruption of the blood-brain barrier. There is a growing interest in effective methods of diagnosing TBI utilizing that serum brain-specific biomarkers as a supplementary techniques, as supplementary methods for MRI and CT. The previous studies have shown that the low heart rate variability entropy values are associated with higher mortality rates. However, that relationship between the heart rate variability, entropy, complexity, and the blood-brain barrier integrity still remains unclear. For this reason, our research aimed to investigate that relationship between brain-specific serum biomarkers and the heart rate variability entropy in the early stage following TBI. The study aimed to describe the group of 20 TBI patients during the first 24 hours after the brain injury. The median age of the group was 53 and 75% of the group were male. The data was acquired from the center TBI data from the study. The patient's condition were evaluated through the Glasgow Outcome Scale Extended that classifies the cases of the brain injury, the measure of the brain injury. 9 out of 20 patients were classified as poor condition as a part of the scale ranging from 1 to 4, and 11 out of 20 cases were classified as good condition as a part of the scale ranging from 5 to 8. The supplementary information about the cases, about the condition of the patients were evaluated through that Glasgow Scale Extended that ranges from 3 to 15, indicating the mild, moderate, and severe states. The term of the entropy itself is linked to the second law of the thermodynamics, which states that the total entropy of the system and its surroundings can either increase or remain constant in any spontaneous process. Therefore, in a human body which is exposed to external factors, there is the continuous state of fluctuation due to homeostasis, and that is why in healthy people we observe high entropy values. Contrarily, the low entropy values are observed in the cases of aging and in the poor condition of the body. In that context, the physiological complexity states that the lower values of physiological complexity are associated with periodic and random dynamics. However, the self-structured dynamics show high complexity. The multi-scale entropy technique developed by Costa et al. Measures the variability with the complexity of changes in the heart rate, and it consists of 2 main stages. The first one is the coarse graining, in which, basing on the time series, the new coarse-grained time series is created based on averaging the data points in consecutive windows of increasing length. Then, for each individual coarse-grained time series in the second step, the sample entropy is calculated, and then the degree of regularity is obtained. Our method took into account that signal of ABP 24 hours after the brain injury. We divided it into non-overlapping windows that were 20 minutes length. Inside of that windows, individual ones, we found the RR intervals, that is the intervals between the individual heartbeats, time intervals between individual heartbeats. And due to that RR interval information, we measured the multi-scale entropy and compared it with the concentrations of the serum biomarkers. And the results show that there is an increase in the serum concentrations, the concentrations of the S100B and UCHL1 concentrations of the biomarkers in unfavorable cases. And that has shown a strong discriminative power with the area under curve above 0.8. In favorable cases, we observed high entropy values, and in unfavorable cases, lower entropy values. And that is correct as comparing to the things that I stated before. But the most importantly, the relationship between multi-scale entropy and the GFAP protein, which indicates the injury of the brain and neuroinflammation, was negative. And to quickly sum up, the multi-scale entropy has its potential in diagnostics in the field of classifying the TBI cases, and especially there is a potential in that relationship between GFAP biomarker concentrations and the multi-scale entropy values, and we will investigate them in the future on larger cohorts. Thank you for your attention. And I will be grateful for some questions.
So I would like to ask you about your own contribution in this work, in this project.
Yes, of course. As I have said before, the data were obtained from the Center TBI project, so I wasn't collecting them directly. And that is also as it comes to the concentrations of the serum biomarkers. So we had the database of that ABP signals and the concentrations of the biomarkers. But the rest of the information, I implemented the multi-scale entropy algorithm. And not only that, because we looked at the different entropies, like approximate entropy, fuzzy entropy, etc. And due to that, we chose that multi-scale entropy. Multi-scale entropy had the most interesting and the most related to physiological conditions, the outcomes. And based on that, I conducted that analysis in Statistica. Yes. Thank you very much.
Dziękuję bardzo za prezentację. I w takim razie zaprosiłabym kolejną osobę reprezentującą pracę numer 6. Mamy chwilowy problem techniczny, więc będzie minutka przerwy w celu naprawy wyświetlania. Dobrze, bardzo przepraszamy za tą krótką przerwę techniczną. Już wszystko działa, więc zapraszam przedstawiciela 6. pracy z listy. Jeszcze sekundkę, bo te informacje nam muszą odwiesić przerwę techniczną. Na czym? Na YouTubie?
Good morning, ladies and gentlemen. My name is Aleksandra Polańska, and I'm honored to present you the work that I've been conducting with my colleagues from Student Scientific Association in Microbiology Department and with our works tutors. The study is entitled Differences in Clostridioides difficile and Clostridium perfringens antibiotic sensitivity depending on the medium used. And to start, I'd like to say a few words about the acronyms that we have on the slide. First one is EUCAST, and that stands for European Committee on Antimicrobial Susceptibility Testing. And that is a group of experts that develop guidelines for susceptibility testing in Europe. And in Poland, we adopted those regulations in 2011. The other acronym is MIC, that stands for Minimum Inhibitory Concentration, and that is the lowest concentration of an antimicrobial that visibly inhibits bacterial growth. But coming back to EUCAST, what's included in EUCAST guidelines? EUCAST guidelines define breakpoints, testing methods, and even media requirements. And those regulations are updated every year. And the last big change was a new medium for anaerobic bacteria susceptibility testing. The new medium is called Fastidius anaerobi agar with 5% horse blood. The aim of our study was to compare whether MIC values differ depending on the medium used. We used Brucella blood agar that was recommended before 2022 and the new one, Fastidius anaerobi agar. We tested that on in Clostridium perfringens strains to determine MIC of penicillin and MIC of metronidazole, also on both media, and in Clostridioides difficile strains to determine MIC of metronidazole using both BBA agar and Fastidius anaerobi agar. The first step of our study was the sample collection. Our study included 12 strains of Clostridium perfringens that were environmental swabs. They were collected in 2017 and 2019 in a hospital in Silesia, and also 73 strains of Clostridioides difficile that were isolated from stool samples from patients with antibiotic-associated diarrhea. We inoculated all those strains, starting with Clostridium perfringens on both FAA and BBA media. We had at first 12 strains of Clostridium perfringens to determine MIC of penicillin on both media. Then we determined MIC of metronidazole, also for Clostridium perfringens. These strains were the same as at the first case. Also tested on both media. And last but not least, we determined MIC of metronidazole for Clostridioides difficile strains, 73 of them, also tested on both media Fastidius anaerobi agar and Brucella blood agar. After that, we inoculated all those agar plates with e-test containing antibiotics for 48 hours in anaerobic conditions. And after that time, we interpreted e-tests according to current UCAST recommendations. And at the end, we conducted statistical analysis using Statistica program. And here are the results of our study. So for Clostridioides difficile, we detected resistance to metronidazole in 3 out of 73 strains on Brucella blood agar, but also in only in 1 strain on Fastidius anaerobie agar. So that makes a big difference. And also, MIC values of metronidazole differed for the same Clostridioides difficile strains. On Brucella blood agar, it was only 1.5 micrograms per milliliter, but on Fastidius anaerobie agar, it was 1 microgram per milliliter. And we conducted Wilcoxon test here, and it showed statistically significant difference. In the case of Clostridium perfringens, we did not observe any resistance to metronidazole, but we detected resistance to penicillin in 3 strains on Brucella blood agar and only in 2 strains on Fastidius anaerobie agar. MIC values of metronidazole for Clostridium perfringens were comparable between media, and median MIC values of penicillin were the same on both media. So here, Wilcoxon test did not show any statistically significant difference. The main conclusion of our study is that the choice of culture medium can really influence MIC results. The lack of statistical significance in the case of Clostridium perfringens may be due to the small amounts of the strains that we tested. But another really important conclusion is that higher MIC values that we receive when testing on Brucella blood agar may suggest a tendency to overestimate MICs, and that can have relevant clinical implications. And lastly, we had a really concerning finding, the occurrence of Clostridium perfringens strains that are resistant to penicillin, which still is a first-line treatment. So that is really important also for clinical situations. And that is the end of our presentation. Thank you for your attention. I invite my colleagues on stage, and we encourage everyone to ask us some questions.
Thank you very much for your presentation. It was very interesting. I would like to ask you, honestly, I have 2 questions. First of them is about your own contribution in this project, and the second, I think more important, is about antibiotics, because at the first step you used 2 of them. Penicillin and metronidazole, yes? And in the case of Clostridium difficile, you use only 1 of them, metronidazole. Why? Could you explain why, for example, you didn't use the second antibiotic in the case of the second bacteria?
Of course, thank you for those questions. At first, maybe I will explain our own work here on this study. So in every step of the way, we were present, we did everything on our own, but of course we asked for some advice from our work tutors. Sometimes it was really hard to divide work because we are all students, and the laboratory work is in the same hours as our schedule at university. But every weekend we divided that work so the bacteria wouldn't have to wait for us, because sometimes it is impossible for that. Actually, every step of the way we did by ourselves, but only Clostridium perfringens strains that were isolated in 2017 and 2019 were isolated by our work tutors. They were frozen, so we needed to unfroze them. But Clostridioides difficile strains were isolated by us last year from stool samples from patients with antibiotic-associated diarrhea. And maybe now I will answer the question about the antibiotics. So For Clostridium perfringens, we used penicillin because that is the first-line treatment, as I have already said. We wanted to check whether there are some strains that are resistant to penicillin. And we wanted to check also metronidazole because in many Polish hospitals, maybe not fully correctly, but metronidazole is still used for both Clostridioides difficile infections and Clostridium perfringens infections. We know that when we treat those infections with metronidazole, it might, the infections may recurrent, they are not, the patient may not fully recover. Yes, thank you for that word. Recover from these infections. But we know that it is still a clinical procedure. But we didn't use penicillin for Clostridioides difficile because Clostridioides difficile have this inherently resistance to penicillin because they produce beta-lactamase, and that would have that would not be a really concerning finding for us because of that. Thank you.
Bardzo dziękujemy za prezentację i za dyskusję. I w takim razie chciałabym zaprosić kolejnego reprezentanta kolejnej pracy. Czy jest z nami na sali? Okej, jest z nami online. Super, dziękuję za udostępnienie.
Dobrze widać?
Wszystko widać, tak?
Super.
Jeszcze jakbyśmy mogli prosić o załączenie kamery, jeżeli jest taka możliwość.
Tak, pewnie, super.
Super, bardzo dziękuję. Proszę tutaj sekundę, zaraz przełączę kamerę i będziesz mógł zaczynać. Zaraz ci
Super,
powiem.
dziękuję pięknie.
Dobrze, możesz zaczynać.
Czy już? Dobra.
Tak.
Good morning, everyone. My name is Andrzej Zuzak, and today I have a pleasure to share with you my study. I represent Scientific Students Association at the First Department of Medical Radiology in Lublin, and I'll be presenting research called Patient Awareness of Radiation Exposure and Protection in Computed Tomography Examinations. So to begin with, I would like to present the overview of my presentation and start. So as we may know, in recent years, the use of computed tomography has been increased rapidly. It leads to significant increase for exposure to patients. While the CT imaging is really a useful tool, we also have to remember about its side effects. There are two potential health risks that include stochastic effects, like increased cancer risk, and deterministic effects like skin burns or hair loss. Although they are rare, we have to always remember about them. While the awareness of radiation safety among healthcare professionals is high, patients still don't understand this topic. So the main objective of our study was to evaluate how much patients know about the radiation exposure during CT scans and their awareness of radiation protective methods. Our goal was to identify the gaps in understanding in the need of better communication between healthcare providers and patients. So to collect data, we created a questionnaire consisting of 24 questions that include 5 questions related to socio-demographic data. The remaining items assessed patients' knowledge and experiences regarding CT scans examinations. Two of the questions were 5-point Likert scale, and the three of them allowed to multiply choice responses. The statistical analysis was conducted by IBM SPSS statistics program. Single choice nominal variabilities were presented using pie charts, and the other ones were presented using bar charts. The multiple choice responses will be shown in tables within frequencies and percentages. All the results I will share with you were statistically significant, and they were used using chi-square test and Cramer's V test. So, first of all, this study was conducted in Medical University Hospital number 4 in Lublin. The amount of the research group was 100, and the data collection was from 9th of January to March 11, 2023. So the first chart, you can see the sex ratio between female and male. And here I show you the age of respondents. The most of them were over 60 years. Our study group varies in the place of residence. And also, to find our findings, we have to ask about the education and occupational status. So, more of the the most of them of the patients had the CT scan before. Only 22 of them didn't have it. And we also asked about awareness of radiological protection and ionizing radiation. And as you may see, most of them know that we have to protect from the rays, but 28% don't know. Also, we asked about the knowledge of ioning radiation risk, and most of them also answered yes. Significant correlation was within age, because most of the younger patients have heard the term of radiological protection. And also in here, no significant age effect, but it was higher among higher educated respondents. We also ask if the people recognize what we can do to protect ourselves from the ion rays. So only 13% could identify all the protection methods which were listed on the questionnaire. And here I present the ratio of the answers. Most of them indicated the lead aprons and thyroid sheets to protect themselves from the radiation. We also ask if requesting and use of the protection was used among them, and 51% indicated yes, but only the 5% of them requested the protection from themselves. The alarming thing is the remaining 95% have never done so. So the key takeaways for presented data is that high CT exposure during the patients have high CT exposure, but the limited depth of radiological safety knowledge. And younger, better educated patients are more aware of radiological protection concepts. Major gaps exist in understanding dose reduction techniques, which are distance and contract use, and we have to emphasize the need for targeted patient education before imaging procedures. And to conclude, patient awareness of radiation exposure and protection during CT scans is insufficient. Less than half of the respondents were familiar with the basic safety concepts, and most associated protection only with lead aprons. We believe that healthcare professionals should take care a more active role in education patients. Simple actions like offering short explanation before the procedures or providing information leaflets or pamphlets could significantly improve the awareness and empower patients about the radiation. Thank you so much for attention, and I'm open to your questions.
Bardzo dziękujemy za prezentację. Czy ktoś z komisji albo z uczestników miałby jakieś pytania do pracy?
I have one small question. It's very interesting work. Thank you very much. Now we have big knowledge about your respondents' answers, but I would like to ask you, what would you like to do with this knowledge? Because now we understand that it's very important to introduce this informations into patients, but what else? What next? For example, are you planning to prepare some instructions, some protocols for doctors or other people who prepare these analysis, these experiments?
So first of all, I think it's really good feedback to the way how the hospital works, how the world works, because based on this data, we can really understand that there's still a lot of things to do. We can always talk to patients and ask them if they understand this, but maybe we have to explain it in better words and focus more on this topic when, for example, ordering the CT scan. But there are various ways we can do it. For example, like posters, maybe also useful. But yeah, it depends. It really depends.
I ask you as a scientist because we scientists sometimes knew better about some problems with, for example, our machines, our vehicles which we used. But how we can introduce this information, how we can give support for our patients. About it. For example, how we can prepare these posters or things like this. Okay, but I think that it's a bigger problem and it's important to work it in the future. Thank you.
Thank you so much.
Tutaj widzę pytanie z widowni, jakbym mogła prosić o przekazanie mikrofonu.
Okay, so I would like to have a question as a person who would go to CT scan. So you have shown that there are multiple factors that can decrease the side effects of CT scans on the presentation. Maybe you can show it if you
Okay,
have it still
wait a
open.
second. Do I have to? Okay, maybe like this. Is it working or not?
Tak, widzimy prezentację.
Tak, tylko wiesz co, chciałbym wrócić. Teraz chyba tutaj. Mhm.
Yes, yes. So my question is how, like, my knowledge as a patient, what can I do with this knowledge about these all factors that influence the side effects of radiation? How can I use this knowledge going with the CT scan?
First of all, you can ask for yourself, for example, a thyroid shield. It also depends on what length of the maybe what part of the body will be tested. I think it also depends on what kind of CT scan you will have. Because, for example, if we use some contrast agents, the exposure can be higher. It really depends on the type of the CT scan you will get, and also about the type of machine you will be tested at. For example, new generation machines are really low dose. They're supplying low dose. So it really depends. But first of all, I think you can ask for some aprons and shields. Thank you.
Okay, thank you.
Bardzo dziękujemy za prezentację i tą owocną dyskusję. Można już zakończyć udostępnienie prezentacji.
Dziękuję bardzo. Do widzenia.
Dobrze, więc pragnę zaprosić następnego reprezentanta kolejnej pracy. Zapraszam tutaj na scenę.
Good morning, ladies and gentlemen. My name is Jakub Kukla. I am the medical student at the Medical University of Warsaw, and I will be presenting to you the work that I conducted with my colleagues from the Scientific Association of Students in the Histology and Embryology Cathedral. The research is about the podophylotoxin and its derivatives, and the promising effects that might be used in melanoma treatment in the future. So to start, I would like to briefly explain to you what podophylotoxin is. It is a substance that was described in 1880 by Valery Podkozowski, who was a Polish-Russian scientist. It's a substance present in the plants of Podophyllum genu. This substance is currently only used in the topical treatment of the genital warts that you can see here. They are caused by the human papillomavirus infection, and they are a problem mostly affecting African countries. The reason for this limitation in the use of podophylotoxin is its high systemic toxicity. So this involves neurotoxic effects, nausea, cardiovascular effects, and that's really a problem for now because it demonstrates antimitotic properties. So it was hoped at the beginning when it was discovered that it might be used in cancer treatment. However, its derivatives such as etoposide are already well-established agents in cancer therapy and are used in the treatment of cancers like small cell lung cancer or lymphomas. And when I mentioned the derivatives, I would like to now focus on the skeleton of the molecule, which is often used as a scaffold for the derivatives. You can see the etoposide molecule here and the teniposide, which is also another active anti-cancer agent. The particle itself has three significant significance regions. Trimethylphenyl region is important because it is taking part in the mechanism of action of the podophylotoxin. So the reason for its importance is that it binds to the tubulin subunits, and thus it actually interrupts the polymerization of microtubules, and this is why podophylotoxin is acting antimitotically. The other important part of the molecule is tetrahydronaphthalene region. This moiety has the hydroxylic group over here at the first carbon atom of the molecule, And this is a very common modification site that you can see over here on these derivatives. The particle is also chiral. It has 4 chiral centers. So we asked, what can we do with this knowledge? And it is a well-established research on our university. And throughout the years, the team developed several derivatives. And the new derivatives that was developed in 2016 is KL3. So it has this moiety attached to the podophyllotoxin, but we wanted to measure and to discover how it acts on the cell, because we know it has a anti-cancer effect that was actually investigated with the T24 human urinary bladder cancer cells. And we know that it is less toxic to non-cancer cells that was found with the research using HACAT cells, which are human keratinocytes. But the new derivative that was obtained is called the AO20. So it is actually obtained through the body P fluorescent particle attachment to the KL3 particle. And in this research, we measured its influence on the B16F10 murine model melanoma cells. It is a well-obtained model for melanoma research. The methods we used are first the Presto Blue viability assay and morphometric analysis. The Presto Blue viability assay was conducted first. We seeded the cells for 24 hours. We incubated them because melanoma cells are adhesive cells, so we need to wait for them to stick to the wells of the plate. After this time, we added compounds with 5 different concentrations. And then after 48-hour incubation, we added the Presto Blue compound. It is a very interesting way of measuring the viability of the cells. And the particle is metabolized by cells that are alive into a fluorescent substance. And then we measure this fluorescent intensity with the spectroscopy machine. In this case, we used Omega machine. And so the more cells are alive, the more the fluorescence, the more strength of the fluorescent signal we receive. Then we analyze the data, and we were able to calculate the IC50, which you can see here is significantly higher for AO20 with 80 micromoles compared to podophyllotoxin, which is, I used this short PDOX here. The morphometric analysis was pretty straightforward. We seeded the cells on the slide, on the 8 well slide. And then we added the compounds after the incubation, and after another 24-hour incubation with the compounds, with the concentrations that you can see here. These are the concentrations used. So we used one for the one lower than the actual IC50. We used also the IC50 and one that was higher. And of course, the control group was also included. After this time, we stained the cells with hematoxylin and eosin with the Harris method. And the cells were analyzed under the light microscope, which I would like to present to you now. This is the control group of the cells. We can see how the shape looks of the cells. So we can see that the cells are in this triangular-like shape. There's a lot of protrusions, approximately 3 to 4 per cell. We can see the nucleoli in the nucleus of the cell, like here or here. And we can see that the shape of the cells is mostly uniform among the whole population. This is the lower concentration of podophyllotoxin. You can see it doesn't affect the cells that much. We can see some of the cells that are slightly enlarged over here. And as we calculated the protrusions, there's a lower amount of them. This is the lower concentration of AO20. It looks very similar to the previous images that I showed you. We can see that the cells have started to kind of gained this needle-like shape over here, with lower number of protrusions. And right now, I would like to present to you what the cells look like after the half maximal inhibitory concentration treatment. So you can see that here the cells are disintegrated in some places. The concentration of the nucleus is also visible over here. Over here, the nucleoli are not that visible anymore. And of course, the population is not as uniform. So we can see some bigger cells. The cells lost are more rounded at some places. There's less protrusions. So that was for the PTOX, and this is for AO20. The cells are in this needle-like shape that I mentioned before that they started to gain. The number of the cells is, of course, decreased. And there's a variability within the shape. We can see some cells that are in this triangular-like shape, some are needle-like shape, and this may indicate the stress occurring within the cells. Just for the comparison, I am also presenting the increased concentration, which we can see acts extremely cytotoxically. We can see the disintegration of the cytoplasm over here. And the nuclei started to deform. So what are the conclusions of this research? We think it's a very important that we discovered the anti-cancer effect that is present within the melanoma cells by the compound, and it was actually similar to other effects of KL3 that were obtained from studies including other types of cells. However, there was no study including B16F10 and AO20 specifically. So we also think that it's worth to compare it with HACAT cells that are non-cancer cells, so that we discover whether the cytotoxicity is lower on the non-cancer cells. And we believe that in the future, the clinical application of this or other derivatives will be possible. That's why it's also important for us to understand the mechanism of action. And this is the key part of this research. AO20 is a fluorescent protein. And by that, it allows us to track it under the confocal microscopy. So we can do the molecular tracking research in the future, which we're planning to do. And thanks to this, we hope to discover how the substance acts and what are its properties. Because right now we're only starting to understand it more. So thank you very much, and I would like to answer any of your questions if you have them.
I would like to ask you about your control group, because in your presentation, it was possible to see that you performed this analysis also in the control group, yes? And I would like to ask, what did you use as a control agent, and which cell line you used as a control line, non-cancer normal cell line, or which cell line would you like to use in the future as a control without cancer?
So in this case, we were aiming to discover the influence on the cancer cells. So the control group were the cancer cells with the medium itself. We used the PEM medium with pyruvate and high glucose concentration. But in the future, because the previous studies conducted with KL3 derivative, so the parenting compound of AO20 were with the HACAT cells, and we know the influence very well because it's also very much described. That's why I think the best option for this study would be to use HACAT cells too in the future, and that's what we're actually planning to do. So the viability assays are important in this case to have this comparison between cancer and non-cancer cells. Thank you.
W razie bardzo dziękujemy za tutaj tą prezentację i zapraszam kolejnego uczestnika.
Okay, so welcome everybody. I would like to show to you results of my small study, effects of partial VGF, VFGF, R2 gene silencing on the expression of metastasis-related genes in non-small cell lung cancer cells. And for the beginning, quick background, why lung cancer? Because it's the cancer with most incidence annually diagnosed worldwide, and the cause of the most cancer-related deaths worldwide. So that's why. And non-small cell lung cancer, it's the most common type of the lung cancer. So that's why this study chose this cancer as a model. And the 5-year survival rates are still very low. For all stages, it's around 25%. So it's really important to look for the solutions for the therapy for this type of cancer. And the aim for this study was to determine the effect of the silencing of the KDR gene on the expression of some metastasis-related genes in A549 cell line as a model of non-small cell lung cancer. And the silencing was performed based on the RNA interference. So the cell was transfected with a specially designed siRNA. And inside the cell, risk complex was formed. This is the RNA-induced silencing complex that degrades target mRNA and inhibits the gene expression this way. So materials and methods. Cell line was A549, as I said earlier. 45,000 cells per well in 6 biological repeats. In 1 milliliters of F12K medium was added to the plate and then incubated so the cells could attach to the bottom. Then the cells were treated with sRNA. Of course, control wasn't treated. Incubated for 48 hours, and then cells were harvested directly with the extracting solution, and then transferred to tubes. And the sequence of RNA is shown here. Next, the concentration of RNA extracts were measured, and the extracts were diluted. Then the RT-qPCR were performed with specific primers, and the list of genes is shown here. We tested 28 different genes. Yeah. And the data from the RT-qPCR was transformed using the comparative method with the fold change of expression. And the results were tested with one sample T and Wilcoxon test. And the statistical analysis were performed using a GraphPath Prism program. And the results are shown here. Word machine is three genes, KDR, our silence gene, TIMP1 and TNC that shown significantly decrease of expression. But there are some other cases where genes weren't significantly different from the control, but we see that something's happening. For example, angiopoietin 1 or tubulin 3 or TIM4. But the next study will be needed to determine if these changes are relevant or not. And for the conclusion, there is correlation between silencing of KDR and two metastasis-related genes, TNC and TIMP1. And follow-up study will be performed to determine other genes if they're really changing their expression or not. And that's it. Thank you. And if you have any questions, I will be pleased to answer them.
I would like to ask you about your contribution in this project.
So my contribution was all the work that was done with the cells, beginning of the putting them into the plates, extracting the RNA, and then performing the RT-qPCR, and then the data analysis too. So I did all the lab work and data analysis. Thank you. Anyone else?
Dziękuję bardzo za prezentację i tą dyskusję. Zapraszam kolejnego reprezentanta kolejnej pracy.
Good morning, everyone. My name is Kacper Jaśkiewicz. I represent Jagiellonian University in Kraków and heart team that focuses on describing the anatomy of the human heart. My work's title is Morphometric Analysis of the Left Ventricular Outflow Tract and its Surroundings in Human Cadaveric Hearts. So, what were the aims of this study? The first and most important aim of this study was to define the anatomical borders of the left ventricular outflow tract. In majority of the studies, authors treat this structure as a plane or as a flat surface, which it is not. It is a spatial three-dimensional structure that has its own height and changes its shape and size during the cardiac cycle. Then we assess surrounding structures of the left ventricular outflow tract and evaluated its dimensional parameters. When it comes to materials, we analyzed 187 hearts from healthy donors with no history of any heart diseases. 16% of them were males. The donors died due to suicide, murders, car accident, etc. The mean age of the donors was 47 years. So let's start with our definition of the structure. On your right, you can see the whole structure. In the superior sector of the photo, you can see the aorta, then the aortic valve with 3 leaflets, And here you can see the anterior mitral leaflet and the structure all around it. It's the left ventricular outflow tract. The most common definition of this structure is that it is a structure located between the anterior mitral leaflet and interventricular septum. Our definition is that it is a structure located below the virtual basal ring, so the line connecting nadirs of each aortic leaflets between anterior mitral leaflet and interventricular septum. And we added definition of the inferior border. It is a line made by edges of the anterior mitral leaflet, and the corresponding points of those of this edge reflected onto the interventricular septum. And you can see those borders on the photo on your left. So it is the virtual basal ring on your on the red color, and on green, in green color, you can see the inferior border. On your right, you can see a photo with pins. It is how I saw a specimen when assessing and measuring different structures within the outflow tract. We also measured the distance from the tip of the anterior mitral leaflet to the virtual basal ring. And the results from these three basic measurements you can see on your left. What is important, all of these measurements varied statistically significantly between males and females. Males had on average greater distances of all of these three measurements. That is not surprising because males have on average bigger hearts and greater dimensions of the chambers within the heart. We also divided the left ventricular outflow tract into three different sectors by lines that divide the intraleaflet triangle, so these structures, into two even halves. These borders you can see in the blue color, and each sector is named after the leaflet located directly superiorly to them. So we have a sub-right coronary leaflet sector, sub-non-coronary leaflet sector, and sub-left coronary leaflet sector. We did divide this structure into those sectors to assess surrounding structures of the left ventricular outflow tract. By penetrating a tissue in the center of the sector, so at the level of the nadir of each leaflet, and at the half of the distance between the superior border and inferior border. And we injected or penetrated a tissue here, and we saw where the needle went to assess surrounding structures of each sector. When it comes to sub-left coronary leaflet sector, in 85% of cases, it was adjacent to the left ventricular free wall. In 14% of cases, it was adjacent to the left atrium. When we measured height in this sector, we found out that when it's surrounded with the left atrium, the height was greater than when it's surrounded with the left ventricular anterior wall. And this is statistically significant. Why? Because if this sector surrounded with the left atrium, the left coronary leaflet was moved slightly towards the anterior mitral leaflet, so away from the commissure that is located here, where the height is the smallest. And that's why we saw these differences. Similar thing happened in sub-non-coronary leaflet sector. So in 79% of cases, it was adjacent to the left atrium. In 20% of cases, it was adjacent to the right atrium. And when it's surrounded with the right atrium, the height was smaller than when it's surrounded with the left atrium. Why? Because when it's surrounded with the right atrium, this leaflet, so non-coronary leaflet, was moved away from the AML, so anterior mitral leaflet, towards the commissure that is here. Here is the commissure of the mitral valve, where the height is smaller. And this difference is also statistically significant. When it comes to sub-right coronary leaflet sector, in 93% of cases, it was adjacent to the right ventricle. In 6% of cases, it was adjacent to the right ventricular outflow tract. But we didn't see any differences when it comes to height in this sector when comparing different surroundings of this sector. And that's all. Thank you for listening.
Thank you very much for your presentation. It was very interesting. Honestly, it was my first contact in my whole life with this small part of our heart. Amazing, very interesting. Thank you very much. But I would like to know where we can apply this information in very simple words.
As I said before, researchers treat this structure as a plane, as a flat surface, which is not. And different dimensions of this structure is different when comparing to different heights. So it is important to know when we measure dimensions of the structure to measure it at the same level. But different researchers measure it on a level 2 millimeters from the anterior aortic valve or 5 millimeters from the aortic valve, and there are no, the definition is incomplete. So I wanted to provide a precise definition so we know what we are talking about when researching or measuring some things in this heart.
Okay, so now we have this information and they are more normalized. We understand them the same in very different places. Okay, thank you very much.
Jeszcze mam pytanie.
To ja mam jeszcze pytanie, czy sposób przechowywania tych preparatów, bo rozumiem, że one były konserwowane w formalinie od donatorów, czy on może wpłynąć na anatomię? Te milimetry mogą robić różnicę, jak formalina wpłynie na strukturę?
Yes, so the specimens were immersed in formalin solution, and we did study the influence of the formalin solution on a heart and different measurements of the heart. And the differences, they are present, but they are very small.
Dziękujemy za prezentację i dyskusję i zapraszam kolejnego reprezentanta kolejnej pracy.
Szanowni Państwo, ja nazywam się Klara Ferenc i reprezentuję Koło Naukowe Katedry Biologii Molekularnej Wydziału Nauk Medycznych w Katowicach. I chciałabym dzisiaj przedstawić Państwu pracę dotyczącą analizy macierzy pozakomórkowej produkowanej przez fibroblasty pacjentów osteogenesis imperfecta. Osteogenesis imperfecta jest chorobą, a właściwie zespołem chorób genetycznych, które dotyczą rodzaju łamliwości kości. Wyróżniamy kilka typów klinicznych tej choroby, z czego za najcięższy i najbardziej letalny okołoporodowo uznaje się typ 2. Jeśli chodzi o podłoże molekularne tej choroby, jest ono stosunkowo słabo poznane. Wiemy póki co, że istnieją pewne mutacje dotyczące głównie genów dotyczących kolagenu typu pierwszego, jednak poza tym wiele aspektów jest wciąż niejasnych. Z tego powodu celem naszej pracy była analiza macierzy pozakomórkowej produkowanej przez fibroblasty pacjentów z osteogenesis imperfecta typu drugiego. I żeby zrobić to w sposób jak najbardziej dokładny, postanowiliśmy wykorzystać modele komórkowe 3D, które w dużym skrócie zapewniają lepsze odwzorowanie struktury architektonicznej tkanek oraz bardziej odpowiadającą warunkom in vitro strukturę macierzy pozakomórkowej. W pracy wykorzystaliśmy fibroblasty skóry, pierwotne fibroblasty izolowane od 3-dniowej pacjentki ze zdiagnozowanym osteogenesis imperfecta typu drugiego oraz fibroblasty pierwotne izolowane od 7-dniowej dawczyni. Pracę podzieliłam na 4 główne etapy. Pierwszym było opracowanie i standaryzacja komórkowego modelu 3D, następnie ocena żywotności komórek w utworzonych sferach, później ocena histologiczna modeli otrzymanych sferoidów, a także jakościowa analiza wybranych elementów macierzy pozakomórkowej. Tak więc zaczęliśmy od optymalizacji i od stworzenia sferoidów z obu linii komórkowych. Na początku wysiewaliśmy 1 000 000 komórek na dołek płytki 6-dołkowej i jak Państwo widzicie, ta liczba była zdecydowanie za duża, ponieważ sfery powstawały w różnym czasie, miały różne kształty, no i było ich generalnie zbyt dużo, żeby je efektywnie analizować. Po wielu różnych próbach doszliśmy do tego, że optymalną ilością będzie 100 000 komórek na dołek płytki 48-dołkowej. W ten sposób byliśmy w stanie otrzymać zwykle jedną sferę, która była łatwa w utrzymaniu i w późniejszej obserwacji. Już na tym etapie zaobserwowaliśmy kilka znaczących różnic pomiędzy sferoidami pochodzącymi z komórek kontrolnych a z komórek SOI, ponieważ sferoidy kontrolne wykazywały bardziej heterogenną strukturę, sfery miały kształty bardziej nieregularne, natomiast sferoidy z osteogenesis imperfecta tworzyły bardziej kuliste, zwarte struktury, które wydaje się, że mogą wynikać z tego, że macierz pozakomórkowa w przypadku osteogenesis imperfecta jest upośledzona z wielu mniej elementów, w związku z czym komórki silniej do siebie przylegają. Kolejnym etapem było sprawdzenie żywotności komórek w otrzymanych modelach. I jak Państwo widzicie na załączonych fotografiach, komórki w zdecydowanej większości barwiły się za pomocą diostanu fluoresceiny, barwiąc się na zielono, co wskazywało dobrą żywotność komórek. Natomiast tylko pojedyncze komórki widoczne w formie takich małych czerwonych hotspotów barwiły się bromkiem etydyny, co wskazywało na komórki martwe. Więc podsumowując ten slajd, generalnie barwienia wykazały, że model był żywotny oraz nadawał się do późniejszych analiz. Kolejnym etapem badań była seria barwień histologicznych. Zaczęliśmy od podstawowego, od hematoksyliny i eozyny. Barwienie to pozwoliło nam na ocenę ogólnej struktury morfologicznej otrzymanych modeli. I tutaj główną obserwacją były różnice w sposobie rozkładania się macierzy w przypadku sferoidów kontrolnych i sferoidów z OI. W przypadku sferoidów kontrolnych macierz była stosunkowo homogenna, rozkładała się w miarę równomiernie, natomiast w przypadku OI miała tendencję do tworzenia pasm, które lubiły lokalizować się na obrzeżach sfery. Kolejne barwienie czerwienią Syriusza, miało ono na celu zbadanie zawartości włókien kolagenowych i tutaj wyniki były bardzo przewidywalne, ponieważ sferoidy kontrolne rzeczywiście zawierały więcej włókien kolagenowych, co jest widoczne jako takie drobne czerwone wtręty, natomiast osteogenesis imperfecta jedynie pojedyncze. Następne barwienie było to barwienie AB-PAS. Jego głównym celem jest wykrycie glikosaminoglikanów, w tym kwasu hialuronowego. Wybarwiają się one na kolor niebieski. I tutaj obserwacja była bardzo interesująca, ponieważ wykazaliśmy, że sfery z osteogenesis imperfecta produkują tych związków zdecydowanie więcej niż sferoidy kontrolne, co jest właśnie widoczne jako ten niebieski kolor, zarówno po 1 tygodniu hodowli, jak i po 2. Podobne tendencje wykazaliśmy w przypadku barwienia orceiną, która wybarwia włókna elastynowe. Tutaj również sfery z osteogenesis imperfecta wydawały się produkować więcej tych włókien elastynowych. Kolejnym etapem naszych badań była analiza jakościowa wybranych elementów macierzy. Tutaj zaczęliśmy od kolagenu typu pierwszego. Wyniki z immunofluorescencji wykazały, że col1A1 występuje w zdecydowanie większej ilości w przypadku sferoidów kontrolnych niż w przypadku OI, co również wykazaliśmy podczas badań immunohistochemicznych. Jest to zgodne z dotychczas poznanym mechanizmem molekularnym i genetycznym tej choroby, także wskazuje, że model dobrze się sprawdził i działa. Natomiast ciekawa obserwacja była w związku z barwieniem COL3A1, czyli łańcucha alfa-1 kolagenu typu trzeciego, ponieważ tutaj zauważyliśmy tendencję odwrotną. Zarówno w immunofluorescencji, jak i w immunohistochemii sferoidy z OI wydawały się mieć większą zawartość tego związku, zdecydowanie większą. No i tutaj postawiliśmy taką hipotezę, że być może jest to wynik tego, że jeśli sferoidy z OI mają mniejszą zawartość kolagenu typu pierwszego, to być może jest to jakaś rekompensata komórkowa poprzez zwiększenie syntezy kolagenu typu trzeciego. I podsumowując, w tych badaniach udało się nam opracować żywotny i optymalny model sferoidalny 3D, który umożliwił badanie macierzy pozakomórkowej w warunkach in vitro, a także wykryliśmy szereg zmian w macierzy pozakomórkowej, fibroblastów z osteogenesis imperfecta typu drugiego. I podsumowując, obserwacje te mogą przyczynić się, mam nadzieję, że się mogą przyczynić do poprawy jakości oraz potencjału badań przedklinicznych OI typu drugiego, a także innych chorób tkanki łącznej, ponieważ model ten, mamy nadzieję, to też będziemy sprawdzać, będziemy spróbować zastosować do badania innych chorób związanych z tkanką łączną. Jednak przedstawione badania jest to dopiero początek naszych analiz, ponieważ jeśli chodzi o najbliższe plany, chcielibyśmy przeprowadzić analizy bardziej ilościowe, przede wszystkim za pomocą techniki RT-qPCR i Western blotów, zbadać ekspresję wybranych, najbardziej kluczowych genów i białek związanych z macierzą pozakomórkową, żeby lepiej zrozumieć mechanizmy, które tutaj zaobserwowaliśmy wstępnie, no i zobaczyć, co dokładnie w tej macierzy u pacjentów z OI typu drugiego się dzieje. I dziękuję bardzo za uwagę. I zapraszam do dyskusji.
Jestem trochę w szoku, bo wytrąciła mi pani z głowy pytanie, które chciałem zadać, tłumacząc, co planuje pani zrobić dalej, więc już za to bardzo serdecznie dziękuję. Prezentacja fantastyczna, naprawdę, bardzo mi się podobała. Chyba nie mam pytań do samej prezentacji, ale chciałbym zapytać, jaki był pani wkład w jej, w przeprowadzenie tych badań. I jeszcze jedna ważna rzecz, jaki był wkład pani w podjęcie decyzji o badaniu w tym obszarze? Czy to była czyjaś sugestia, na przykład opiekunów, czy to raczej wynika z pani osobistych zainteresowań?
Więc odpowiem na oba pytania za jednym razem, ponieważ mój wkład własny było wykonanie właściwie większości tych, tak naprawdę wszystkich prac laboratoryjnych. Oczywiście to było robione w ilość powtórzeniach, więc nie powiem, że wszystkie z 5 powtórzeń wykonywałem ja, ponieważ czasami zdarzało się tak, że faktycznie ktoś z moich opiekunów wykonywał te barwienia, więc pracowaliśmy zespołowo. Natomiast w każdym etapie tych prac uczestniczyłam, zarówno w optymalizacji tej hodowli, w zastanawianiu się, jakie płótki, w jakiej ilości komórek robić, które barwienia, później interpretacja, samo przeprowadzanie tych analiz histologicznych, barwień immunofluorescencyjnych, immunohistochemicznych. Natomiast etap, którego nie robiłam i nie robili też moi opiekunowie, była to część dotycząca przygotowywania szkiełek, taka czysto techniczna na mikrotomie, ponieważ to robiła dla nas katedra histologii, także szkiełka były im przekazywane, następnie brane i później odparfilmowane i barwione dalej już u nas w katedrze. Natomiast drugie pytanie, nie wiem jakie w sumie było, więc jakby mógł pan przypomnieć.
Skąd pomysł na tego typu badania? Przez sekundę też musiałem je sobie przypomnieć.
No właśnie, więc generalnie osteogenesis imperfecta jest to choroba, która w katedrze biologii molekularnej jest już, że tak powiem kolokwialnie, męczona od wielu lat. Tak też korzystając z możliwości dostępu do takich komórek i możliwości po prostu wkręcenia się w tą tematykę badawczą, z niej skorzystałam. Natomiast jeszcze chciałam wrócić do wcześniejszego pytania, którego końcowo pan nie zadał, natomiast miał pan na myśli. Poza tymi rzeczami, które tutaj wypisałam na prezentacji, chcielibyśmy właśnie rozszerzyć też te badania na inne choroby tkanki łącznej. I ponieważ kiedyś w naszej katedrze były prowadzone dotyczące tętniaków aorty brzusznej, mamy również zgromadzoną pewną pulę komórek, również tkanki łącznej, również fibroblastów, z których właśnie w najbliższych miesiącach chcielibyśmy spróbować wyhodować podobne sferoidy, żeby zobaczyć, jak te zmiany, czy możliwe są do zaobserwowania w warunkach in vitro, jakie zmiany z kolei właśnie w tętniakach aorty brzusznej. Więc też, mimo że prezentacja dotyczy dzisiaj osteogenesis imperfecta, to tutaj skupiam się i na aspektach technicznych dotyczących samych sferoidów, ponieważ jest to technika, która dopiero w ostatnich latach tak naprawdę zaczęła być szerzej stosowana w badaniach podstawowych. Oczywiście pojawiła się dawno, natomiast na większą skalę tak naprawdę dopiero od niedawna zaczyna się wykorzystywać. No a po drugie dalsze właśnie analizy macierzy już OI-ów, o których tutaj już wspomniałam na slajdach. Dziękuję bardzo.
Bardzo dziękujemy za prezentację i za tą dyskusję. W zapraszam kolejnego przedstawiciela kolejnej pracy tutaj na scenę.
Good morning, everyone. My name is Victor, and I'll be presenting our research in anti-cancer effects of curcumin. So, what exactly is curcumin? Curcumin is the main polyphenolic active compound derived from turmeric or curcumin, curcuma longa. It's the thing that gives the turmeric its characteristic orange-yellow color. Curcumin is widely studied for its therapeutic use in anti-inflammatory, antioxidant, and anti-cancer effects. And colorectal cancer is CRC is one of the most common cancer in the world. Unfortunately, it is diagnosed late, so it leads to metastasis or making curing it much more difficult. But curcumin helps here because curcumin presents some anti-cancer properties like inhibition of cancer cell proliferation. It stops cell cycle arrest, induction of apoptosis. It inhibits angiogenesis and metastasis. It has anti-inflammatory effect, and last but not least, a safe profile, which makes it potential in prevention and supportive therapy. And for our research, we use two colorectal cell lines. We used HT29 and CACO2 cells. Both were derived from patients with colorectal adenocarcinoma. And we performed some sequence of our, this is a sequence of our test. First, we treated our HT29 and CACO2 cells with curcumin at various concentrations, ranging from 6.25 up to 200 micromolar. Next, after incubation, we performed WST1 assay, which assessed our cell viability. Next, WST1 assay was performed after 72 hours. Just like crystal violet staining, also after 72 hours was performed to assess the total number of adherent cells. Next, to visualize nuclear changes and detect apoptotic cells, DAPI staining was performed after 48 hours of incubation. And finally, we also performed a wound healing assay, which was used to assess the effect of curcumin on cell migration. So first, we will look at the effects of our results for the WST1 assay for HT29. And we can see that dose-dependent manner here shows that curcumin affects cell viability with higher doses of curcumin, with high concentrations. This cell viability decreased. And this same pattern we can see in CACO2 line, because as well, dose-dependent manner in cell viability, and in higher concentrations, higher the degrees of the cell viability is lower. So next we had crystal violet staining. It was performed to show us how much adherent cells we have after the treatment. We can see as well, like in the WST1 assay, that in HT29, in higher concentration of curcumin, we have less adherent cells. So The same we can say about CACO2 cells as well. There are less adherent cells. Next, we performed DAPI staining, which we used to see changes in the nuclear morphology and to see changes. DAPI stains to the nuclear and we can see that these red arrows show us cells in apoptosis. It shows us that the same results we can see in HT29 and in CACO2 cell lines. It shows us that in higher doses of curcumin, that higher doses of curcumin not only lower cell viability, but as well it induces apoptosis in those cells. Next, we performed wound healing assay that showed us migratory effect of curcumin. We can see that in control groups, which was untreated cells, the wound was the cells almost cover all of this scar here. But in the cells treated with curcumin with 50 micromolar concentration and 200 micromolar concentrations, the gap stayed really wide. And after 48 hours, we can see that in the control group, this scratch is almost completely covered in cells, but still in the 50 and 200 curcumin concentration, the gap still remains. The cells really slow affects this gap. The migratory effect of these cells are very are lower than in the untreated cells. And as well, the same effect we can see in the CACO2 cell line, because in the control, the scratch is covering slowly, but we can see it is better seen in the 48 hours. After 48 hours, because we can see that the scratch is completely covered, and these higher concentrations, it shows that in the high concentrations, the migratory effect of the cells are lower. And now, to sum up, potential of curcumin in the against HT29 and CACO2 cancer cells lines shows that we can see that from our research that curcumin affects cancer cells not only on inhibitory, but as well in cell proliferation and migratory capacity. And it is promising for us on the working with curcumin at the cancer cells in HT29 and CACO2 colorectal cancer lines. Thank you for your attention. So there are any questions for me or us?
Okay, so can actually can a doctor prescribe me some curcumin for like cancer? Because it seems like okej, z tego co zrozumiałem, czy to jest jakby faktycznie możliwe, żeby doktor przepisał mi kurkumę jako...
Not yet, because right now curcumin is still under studying. Right now curcumin is only as a diet
supplement of diet. So yes, you can supplement it, but it's not yet confirmed as a drug. So just a supplementation right now.
Okej, dziękuję.
And I would like to ask about the differences in response to curcumin between two cell lines, HT29 and CACO. So what is the cause this difference? Because HT29 was more chodzi mi o to, że większy efekt był widoczny tak na pierwszy rzut oka na HT29 niż na KAKO. Skąd ta różnica może wynikać?
So the difference can be on our side because the difference is not so big as we can see. It is here, but it is not so big. I think it might be our thing because maybe we
because all these laboratory tests we did by ourselves, so it could be maybe a human layer factor, but yeah, we just think like that. Honestly, we don't know, just so we don't have this information. Yes.
Okay, so I want to ask you if we should take curcumin as a diet supplement, even if we don't have any disease, and if yes, what dose per day, for example, should we take?
We could, but it doesn't have. It's still under studying. Yes, we still don't have so much information about that. But right now, these diet supplements are dosing in one tablet. Yes. So as I know, the one tablet for a day is good for now. But as I said, it is still under research. How many? I mean, we still don't exactly know the effect, the cancer effect. We still don't exactly know everything about curcumin.
Okay.
Just we need research more.
Thank you
Yes.
so much.
Thank you very much.
Dziękujemy bardzo za prezentację, za tą owocną dyskusję i zapraszam następnego reprezentanta następnej pracy.
Okay, so I'm from Wrocław, from Wrocław University of Science and Technology, and I'm going to show you how signal processing in our project called RoboWapa on myoelectric hand prosthesis works. Okay, so here's our project, our prosthesis. First of all, we designed it, then we assembled the hardware, the electronics, then we figured out how to acquire MG signal and how to analyze it, and then we programmed it. Okay, so the MG signal processing. So basically, first of all, we have to acquire it. We acquired it through electrodes. Then we process it in code and with the usage of malware. Then we tried many strategies how to actually translate NG signal to hand movements, and then we applied it in our processes. Okay, so as you may see, efficiency hides in simplicity. Here's our electronics. We have one Arduino, one malware sensor, three standard servo mechanisms, and two continuous servo mechanisms. So basically, here's our code. It's like the way our hand thinks, let's say. First of all, we read signal. Then we assign maximum value. We do that because malware gives us values from 0 to 1024. 0 for like relaxed muscle, 1024 for uh, uh, contracted muscle. And you may ask us, okay, but why do we do that? So basically, we have to know what's the max sensor value, or how strong can a person contract their muscle, to know when a hand should close and when it should open. Then here's our closing and opening logic. We basically check if current sensor value is bigger than max sensor value divided by 2, and if hand is opened, exclamation mark before closed means like the opposite of closed. So if it was opened, then we open the hand, then we close the hand, and else if it was closed before, we open it. At the start of our program, we have the initial calibration. So we just ask the user to contract and relax their muscle to check what's the maximum value of the contraction. And then we have a delay to allow smoother movement. Okay, so what exactly are servo mechanisms? Servo mechanisms are like small motors that move from 0 angle to 180 in standard type, or move continuously in continuous rotation type. But in continuous rotation type, the coding is quite trickier because for values from 0 to 90, it goes all the way left. And from values from 90 to 180, it goes all the way right. And it stops at 90. Okay, so what are the pros of our RoboHand? Basically, it's incredibly simple and it's lightweight. It's 3D printed, so basically it weighs around half kilo because it's quite big, quite large right now. Moreover, our calibration adapts to the user. It constantly checks what's the maximum value of the muscle contraction is, and then just adapts to that. Also, at the startup, we filter the values. That's the whole calibration thing. For the first 5 seconds, hand doesn't move. It just reads the values from the user. And it's quite fast, runs incredibly smooth, but limitations. So right now, our hand can only close or open. There's no in between. Like, there's no, I don't know, fancy gestures or anything. Moreover, there's a single threshold for closing and opening. So let's say someone will not reach this threshold again, despite many tricks we used in signal analysis. It's still, it can sometimes trigger on like false triggers. Speaking about false triggers, we have no hysteresis. Basically, sometimes hand may rapidly close and open because of the fact that because of the artifact in malware 2.0. And moreover, like I just said, unstable maximum calibration. Basically, AMG spikes may incorrectly redefine the range. Let's say someone has 700 on muscle contraction, and then randomly it spikes, and then it's quite hard to use it. So what's our future outlook? We aim to make it more lightweight, which is quite doable. Moreover, we want to make our code better, make it more adaptive to the user, and make it more cost effective because right now it's not as costly as many processes. It's around 500 zlotys, but we want to make it even cheaper. And our future plans, as I said previously, it's quite heavy. We want to make it weight even less. Our literature. So thank you for your attention. And my colleague's going to. Okay.
So you mentioned that there is a single threshold right now that is implemented, and I just wondered, because you didn't mention in your future plans if you're trying to do something about this, so maybe you already thought about the way to kind of differentiate between the EMG signals or another solution to this problem so that it can be actually more adaptive.
Okay, so of course I have quite a lot of ideas. Most of them I think wouldn't work unfortunately in real life. So for example, one of them was to make it calibrate every time, but I think that it would make the robot hand more like clumsier, like the movements would have larger delay, and moreover, so the one thing I thought about was actually checking only 5 latest max signals and then kind of resetting. But that doesn't seem like a good idea because if someone just doesn't contract their muscle, it just zeros.
Thank you.
W razie bardzo dziękujemy za dyskusję i za prezentację. I zapraszam kolejnego przedstawiciela, reprezentanta kolejnej prezentacji.
Dear committee, dear guests, dear participants, my name is Magdalena Szustkiewicz, and today I would like to present the course and results of a study entitled Analysis of UV Spectra of Energy Drinks and Consumers' Perspectives, conducted in collaboration with and under the supervision of Dr. Jakub Adamczyk. The aim of the study was to analyze the composition of various energy drinks to detect differences between them, An attempt was also made to investigate consumers' habits and preferences. And the research issue under the consideration was to a verification of composition of energy drinks to detect differences between them, which might be related to their increased consumption, which appears to be significant due to the high level of interest in this area. The method employed was UV spectroscopy. Each sample of an energy drink was placed in quartz cuvettes, 10 milliliters each. Measurements were repeated 3 times, and then the results were averaged. During the study, the absorbance was measured and spectra generated by absorption of UV radiation were recorded. The selected energy drinks are actually listed on the slide. And then subsequently, based on analysis of UVs, based on ingredients of energy drinks, their capability of for radiation absorption within a specified wavelength range, comparison of compositions of tested energy drinks, and comparison of maximum absorbance values achieved for each energy drink. An attempt was made to analyze the obtained results. And also for a survey study, a CAVI method was employed, and 70 respondents participated in it. Here are the samples used for the study, with a control sample shown on the left and the test samples exemplary shown on the right. And visual identification of energy drinks was impossible due to the similar color resulting from addition of dyes. And therefore, detecting compositional differences between them was impractical, justifying application of UV spectroscopy for this purpose. And on this figure, spectral absorbance for each energy drink was plotted using different color, and also this slide shows maximum absorbance values, and the preliminary conclusions are as follows. All of the tested energy drinks achieved a maximum absorbance within the ultraviolet radiation range. The absorbance under the same conditions increases with the concentration of a specific component. So drinks like Red Bull Black, Fire Up, and Monster exhibit very similar compositions, raising a question of whether taurine or applied regulatory agents might affect study results. And also Tiger Placebo, a drink. Maximum absorbance value is really close to that of Red Bull, despite absence of caffeine or taurine in its composition. And due to the lack of definitive answers, an analysis was conducted regarding the radiation absorption capability of all the listed ingredients in the energy drinks composition to detect which one of the substances influenced the observed maxima. And after the analysis, substances differing between Red Bull and Black weren't detectable at the obtained absorbance peaks. Therefore, the substances most probably responsible for those results are caffeine, vitamin B6, or vitamin B12. Other ingredients absorb radiation at the wavelengths not corresponding to the obtained results. Regarding energy drinks, the Grape, Fire Up, Monster, and Black Cool Apple substances probably most likely possible for the maximum absorbance were caffeine, apple juice from concentrate, and ginseng extract. And moving on to the survey results, about 90% of respondents reported feeling fatigued through the week. Then regarding the reasons for consuming energy drinks, about 59% of them indicated studying as a primary motivation, with less frequent association to, for example, work. They prominently preferred sugar-free drinks, paid attention to vitamin and caffeine content. Also, a flavor was one of the crucial selection criteria. and the effectiveness ratings of different energy drinks varied widely among consumers. The time after a consumption when the energy drinks effects were felt most often ranged from 5 to 30 minutes mainly, and the duration of effects ranged from 1 hour to an entire day. Moving on to conclusions. First, I would like to focus on energy drinks which were most frequently chosen by respondents. So concerning Red Bull and Black, the substance probably most likely responsible for the maximum absorbance is a vitamin B6, but the differences in results may indicate varying vitamin B6 concentration, which aligns with the manufacturer's declarations, because Red Bull should contain 4.5 milligrams per can and Black 1.75 milligrams. According to the survey data, it is highly relevant as respondents reported paying attention to vitamins content, and the study results confirm the accuracy of composition data. Furthermore, it can be concluded that each of the tested energy drinks, apart from Tiger Placebo, is characterized by a different caffeine content, despite probably almost uniform declarations by manufacturers, because all of them should contain 80 milligrams per can, except G-Grape, which should contain 100 milligrams. And indeed, in comparison of the absorbance at wavelengths characteristic for caffeine, a G-Grape exhibited the highest levels, indicating the highest caffeine content. And however, it is concerning that other energy drinks, despite almost uniform declarations, there were still some differences between them. And according to G-Grape, the survey data revealed that it was not most frequently chosen energy drink, despite having the highest caffeine content. And about 11 respondents selected it as the most effective. In contrast, the energy drinks with the lower caffeine content, based on absorbance analysis, were more frequently chosen as more effective. So it raises a question of whether psychological factors like community or a brand name may influence the consumer perceptions. And the applied study method allowed for the identification of individual energy drinks based on their UV spectral properties. And thank you for your attention.
I have, thank you very much for your presentation. I have a few comments, especially connected with the UVV spectrum. Can we return into this spectrum on this slide? Because I would like to tell you, oh, here, thank you very much. I would like to tell you that, according to my opinion, these dilutions are not enough, because as we can see here, the value of absorption, value of absorbance here is very, very high, is, according to my opinion, too high, because if you would like to get valuable information about the concentration of ingredients in the mixture, we usually use the concentration not high when the absorbance is not higher than 1.0. It's the best ratio between 0.1 to 1.0 is the best ratio, because here we can observe this absorbance, and when we have higher signals we can get only information that here we have absorbance, here we have something inside, but we cannot understand what. So if I can suggest something here, if you would like to diverse energy drinks using UVV spectroscopy, you should dilute these compounds, these products, and obtain the highest absorbance something around 1.0. It should be the highest level. Okay, and thank you very much for your next part of this presentation because it was very interesting. And I would like to ask you about your own contribution.
Well, I participated in every step of this study. Actually, I think that the moment when measurements were done, probably I've just got support from my supervisor because he probably knows better some things I'm like still learning. And if it comes to results, analysis, conclusions, I actually made it by myself. So that's like that.
Fantastic. Thank you very much. Of course, we are still learning, and I have one small suggest for you. You told us that it was very difficult to analyze the concentration of caffeine in these some substances because we couldn't see the band connected with the caffeine because it's close to 217, 275, yes? And here we cannot see peak in this place. But if we will dilute this solution, maybe we could see something in this area. So it could be advice for the future, but now very good presentation, very good work. Thank you very much.
If I could only just add something, we were checking the absorbance within this range where caffeine is actually observed, and that's why we formed those conclusions according to this.
Okay, so I have one question. Have you considered the price of the energy drinks in like preference of the people, or it was a question that was regardless of the price? So if they were to choose, they got for free those drinks, which one they would choose?
In this survey, we actually asked about different things which could have an impact while the respondents were choosing their drinks. So the price was one of the things in this specific question, but most likely was flavor chosen as this criteria which is the most crucial for them.
Dziękujemy bardzo za prezentację i za tą owocną dyskusję. Droga komisjo, teraz najprawdopodobniej wrócimy do pracy numer 2. Is there a represent of the work molecular mechanism of hemophilia A at inversion of intron 22 on Zoom?
Yes, hello.
Hi. Please show your presentation and turn on your camera. Thank you.
Should I share the screen or you will share it?
You share the screen.
Okay. Just a second. I will try to quit and reopen to see if it will work because I'm not sure. Just a second.
We don't see any presentation.
Yes, that's why I will try to.
Okay. Okay. We see your screen. Sorry, we don't hear you.
So now it's okay, I think. Good afternoon, dear jury, dear participants, dear organizers. Excuse me once again for the delay. My name is Trifon. I'm a sixth-year medical student at Medical University of Sofia. And today I'm going to present you our research about molecular mechanisms of hemophilia A at patients with inversion of intron 22. Firstly, a brief introduction about hemophilia A. As we all know, that's the most frequent and severe hereditary bleeding disorder, which is X-linked disease and is characterized by quantitative or qualitative deficiency in the blood coagulation factor 8. There are over 80 mutations in the F8 gene, but approximately between 40 and 50% of patients with severe form of hemophilia A are affected by gene inversion of intron 22. That's why we choose to investigate exactly this mutation in our research. Firstly, the synthesis of a normal factor 8. As you can see, that's the X chromosome in the part of the F8 gene. That's the transcription translation, and as a result, a normal factor 8 is formed, which has 5 domains, as you can see here, 3 A domains and 2 C domains, C1 and C2. As a result of inversion, which can happen in 2 mechanisms, that's the first one, and as you can see here, exactly intron 22 till 26 are missing, and that's the second type of inversion which can happen, and as a result the synthesis is altered and an abnormal protein is formed, and as you can see the C2 domain is absent while the C1 domain is changed. It's reduced with 32 amino acid residues and the last 16 amino acid residues are substituted. In other words, we have 16-point mutations. And as a result of this mutation, the intrinsic pathway of hemostasis is blocked, and that's result in bleeding disorder. What's the aim of our research? Firstly, we aim to reconstruct the three-dimensional structure of inverted factor 8 because this structure is unknown till now. Using its primary amino acid sequence, we also aim to compare the thermodynamic stability and the electric properties of both proteins, normal factor 8 and factor 8 with inversion of intron 22. And finally, we want to investigate the interaction of normal and inverted factor 8 with their plasma carrier, von Willebrand factor, and with factor 9, which is important in the blood coagulation cascade, as I showed you in the previous slide. What we use, we reconstruct the 3D structure of inverted factor 8 using a computer program for in silico mutagenesis. This is the program DUET. After that, we compare the electric properties of both proteins using another computer program for protein electrostatic, which is called PROBCA, and we calculate the thermodynamic stability using the program BOOS, and at the end, we calculate the association energies of normal and inverted factor 8 with factor 9 and von Willebrand factor. Our results, that's the structure of C1 and C2 domain. In the above row is of the normal factor 8, in the below row is of the mutant factor 8. As you can see, that's the C2 domain which is absent. That's the 32 amino acid residues which are absent in the C1 domain, and that's the 16-point mutation in the C1 domain. That's the three-dimensional structure. As you can see here, the C2 domain is absent in inverted factor 8, and the C1 is altered. It's with 32 amino acid residues shorter, and 16 amino acid residues are changed. The electric properties, as we know, at low pH, when the proton concentration is high, the protein is positively charged, which is determined by the proton associating groups of lysine, arginine, and histidine, and that's why the net charge of the protein is positive, as you can see here on the left side. At high pH concentration, when the proton concentration is very low, the net charge of the protein is determined by the proton dissociating group. That's the carboxyl group of aspartate and glutamate, and that's when the net charge of the protein is negative. The pH at which the net charge is 0, which means that the number of positive amino acid residues are equal to the number of negative charges, is the isoelectric point of the protein. And in this situation, as you can see, as a result of the inversion of intron 22, the isoelectric point of the mutant protein is shifted. As you can see, the normal protein has isoelectric point 8.53, while the mutant protein has isoelectric point 7.45. In other words, almost 1 pH unit difference between the two isoelectric points. And as you know, when we have a pH of the medium, which is below the isoelectric point, the protein is positive charge, while when the pH is higher than the isoelectric point, the protein is negatively charged. And in this situation, at physiological pH, which is 7.4, the mutant protein will be almost zero. The net charge will be 0.4, while the normal F8 protein is positively charged, +9, as you can see here. When we speak about the thermodynamic stability, it depends on 4 types of forces. That's hydrogen bonds, electrostatic forces, van der Waals forces, and hydration energy. In this situation, we calculate the electrostatic component of the folding free energy because this is the only one component which is pH dependent. And when we make a graph of the dependence of the folding free energy from the pH, we can see the pH interval in which the protein is maximal stable. The higher the negative value of the folding free energy, the more stable the protein is. And as you can see, both proteins are stable in the same pH interval almost. Normal protein 8 is a little bit more stable, but when we see at physiological 7.4 pH, we see that the normal factor 8 is almost twice as stable as inverted factor 8. The folding energy is approximately minus 120, while of the mutant factor 8 is minus 60, twice as stable. And finally, we calculate the association energy and the dissociation constant of the complexes of normal and inverted factor 8 with von Willebrand factor, which is the plasma carrier of the protein. That's the model, as you can see, of normal factor 8 and of mutant factor 8. That's the 2 domains of von Willebrand factor. As you can see, the main interaction is between the C1 and the C2 domain. And as I mentioned, and as you can see, in the mutant factor 8, the C2 domain is absent, the C1 is altered. That's why we have a change in the association between the two proteins. And as you can see, the higher the dissociation constant, the lower the association is. In this situation, you can see that the inverted factor 8 dissociation constant is approximately 7,600 higher, which means that the association between the inverted factor 8 and von Willebrand factor is 7,600 times lower. When we see the interaction between factor 8 and factor 9, the interaction is mainly between the A2 and A3 domain. These both domains are not altered in the mutant factor 8. That's why the association between the two proteins is not affected. So, in conclusion, inversion of intron 22 of the F8 gene results in the formation of an imperfect protein, and the association of this protein with factor 8 is not affected, but the association of inverted factor 8 with the factor von Willebrand, which is the plasma protein carrier, is affected, which results in shortened circulatory half-life and strongly reduce the plasma concentration of the complex, and therefore patients with this mutation have severe form of hemophilia A. With this, I want to thank you for the attention, and if you have any questions, I can answer.
Thank you so much for your presentation. Now I would like to invite you all to ask a question and discuss the presented paper.
Thank you very much for your presentation. I would like to ask you about your own contribution in this project.
All calculations, all models, all tables, and all figures are made personally by me. Only my tutors help me with some interpretation. But all calculations, all computer programs are used by me because in the another researches which we done before this, Dr. Christova and Professor Zhivkov teach me how to work with these programs. And now that's, so to say, my first own research, and all calculations and everything is made personally by me.
Okay, thank you so much.
Thank you.
Wszystkie zgłoszone prace zostały zaprezentowane. Gratuluję wszystkim autorom oraz uczestnikom naszej sesji nauk podstawowych, biotechnologii i inżynierii biomedycznej. Niezwykle mi miło, że tyle osób zgłosiło się do tej sesji. Jak sami wiecie, w tym roku mamy nawet drugą część, więc to pierwsza taka w historii sytuacja. Więc naprawdę bardzo dziękujemy Wam wszystkim za zgłoszenia i pamiętajcie, że samo to, że już tutaj zaprezentowaliście swoje prace, jest w pewien sposób wygraną. Chciałabym oczywiście ogromnie podziękować jury i tutaj Państwu, którzy są stacjonarnie, i Państwu, którzy są zdalnie i wypełniali karty ocen. To naprawdę dla nas zaszczyt, że mogliśmy Państwa tu gościć i jeszcze raz bardzo dziękuję. Na koniec chciałabym Was serdecznie zaprosić na w sumie 3 rzeczy. W pierwszej kolejności na oficjalne otwarcie i panel dyskusyjny, które odbędą się o 13:00. Mamy ciekawych gości. Będzie pani profesor Misia Stec oraz 2 naukowców z Polskiej Akademii Nauk, więc naprawdę warto. Oczywiście coś, co myślę, że Was najbardziej interesuje, czyli ceremonię wręczenia nagród, która odbędzie się 9 maja o godzinie 17 w auli A3, tak samo jak ceremonia otwarcia. Oraz coś już mniej oficjalnego, czyli After SIMC w DSO o godzinie 21 również 9 maja. Więcej informacji oraz formularz zapisów można znaleźć na naszym wydarzeniu na Facebooku Carlos from SIMC 2025. No i jeszcze taka mała wzmianka przed aulą A3 będzie czekał na Was poczęstunek, więc po tej długiej sesji zachęcam Was do skorzystania. W imieniu Studenckiego Towarzystwa Naukowego chciałabym Wam ostatni raz bardzo podziękować i oficjalnie zakończyć sesję podstawowych nauk biomedycznych, biotechnologii i inżynierii biomedycznej.